Hello, my name is Kristen. I will be discussing how to model molecular reactions using a system of ordinary differential equations. So here are kind of an example of 5 different types of chemical reactions. When you first see this, I think you would initially think of reactions between chemical components, but they can also be used to model different reactions between molecules, like protein interactions that are involved in maintaining, that are involved in driving cell function. Ordinary differential equations are a frequently used tool to model these reactions since they are continuous, nonlinear, and dynamic systems. One of the nice things is that the final output are based on parameters and initial conditions specified by the user, so they can be highly modified based on whatever you're studying. And there are also many programs available that can solve these ODEs numerically, like Python and MATLAB. In this example, I will be showing how to model the assembly of extracellular matrix in response to different substrate stiffnesses. So we know that cell migration actually relies on ECM assembly. To begin with, cells can synthesize fibronectin as a scaffold you can also think of it as like a railroad track that they can use to move along. This is then degraded over time and replaced by collagen to form mature ECM that we typically think of. And here is an example of how this occurs, where the fibronectin binds to integrins on the cell surface, are stretched out, and then allows for this collagen to start binding to it. This is typically seen in wound healing, and here's an example where you have this initial increase in fibronectin in response to this wound that degrades over time, and then after this initial increase in fibronectin, you'll start to see this increase in collagen type 1 occurred and replace this fibronectin. We also know that this rate of ECM assembly actually changes in response to stiffness of the surrounding environment. So the actual stretching of the fibronectin is dependent on the substrate stiffness. This is typically researched in a lot of disease models like tumorigenesis and fibrosis that are both characterized by the change of stiffness of the tissue. This model that I made is built off of 3 different papers. 2 of them are previously made computational models that really look at fibronectin interactions with the cell. And then this third one is a paper that describes the different properties of collagen, such as its disassembly and assembly. The goal of this model that I've made is to incorporate this fibronectin collagen interaction to really see how this ECM, the interactions between these 2 proteins, can change under different stiffness conditions. The previous literature was used to really get parameters for the model, like the initial conditions, reaction rates, and a lot of the constants like the stiffness conditions that were used. And this really shows that you can build off of either previous math models to improve the accuracy, or you can use experimental data as the main driver of your computational model as well. So here's the overall reaction model shown as a diagram where you have this initial interaction between initial binding with the integrin and fibronectin. This fibronectin is stretched, which allows for other fibronectin to bind to it. Then you see this collagen binding, and I also include a fibronectin degradation reaction to fibronectin degradation reaction as well. As you can see, there are 7 different reactions that occur. So the next step is to derive the overall reaction formulas. So there are going to be 1 equation for each interaction in the signaling cascade, and for this specific model, we are associating an interaction as either a forward or reverse reaction. So I have 10 different equations in this model. For For example, there are two equations that represent the different interactions between fibronectin and integrin, the binding and unbinding of them. You can then use these to convert it to a system of ordinary differential equations. Each ordinary differential equation represents the change in concentration of a reaction component. And in this specific model, there are 7. I define changes in the state of each component as a separate variable. For example, there are 2 variables that for integrins. One is looking at free integrins and 2 is looking at bound integrins. The general ODE form is going to take the form of change in the concentration over time is equal to the rate that it's created minus the rate that it's destroyed. And here is an example of the ODE for free integrins, where there is a creation rate based on the unbinding between integrins and fibronectin, and a destruction rate based on the binding between integrin and fibronectin. And another example is stretch fibronectin as well. And you will do this for each of the 7 variables you want to look at. So to solve this, I use Python using the Scipy library and ODE int function. And to define these ODEs, you need to first define any parameters that you're setting, constant parameters you're setting. So for this, I apply the stiffness conditions as a constant variable known as k sub, and then I define the different variables, different parameters that are dependent on the stiffness variable. You'll then define the rate constants for each of the reactions, and then the variables you want to look at. Finally, you'll define the ODEs based on how you define each of these variables. So calling this ODE function is very dependent on user setup. So you yourself will have to specify the initial conditions of each of the variables you're looking at, and also the time that you want to run this ODE over. So it's very reliant on user setup. It's very important to always double check your work while you're creating this function. But overall, as you can see, I modeled 2 different stiffnesses, 0.1 piconewtons and then 1,000 piconewtons. And this is based off of the 2 stiffnesses studied in the Weinberg and Lemon paper that I referenced. As you can see, the rate of ECM assembly is faster on high stiffness substrates compared to lower. You can see this immediate increase in integrin binding and fibronectin binding as well. And another thing that's important to note is that this collagen assembly is also reliant on when the stretched fibronectin begins to occur. So this is also like what is seen experimentally, like what I showed you before, where you see this increase in fibronectin and then the steady increase in collagen as well. And this is definitely a good example of how you can use a system of ODEs to model molecular interactions that really drive cell function. There are also things that can definitely be improved on this model, for example, including synthesis and degradation terms for collagen and fibronectin, because we know that there will be continuous turnover in these proteins in an actual tissue environment, or in the actual tissue. And another thing that could be incorporated to improve accuracy is incorporating this collagen integrin binding as well, because we know that collagen can bind to cells independently of fibronectin, and it might independently of fibronectin. But yeah, and these are the references that I used, and thank you for listening.